10 past 11. Welcome guys, good morning, sanbonani, dumelani, avsheni, hello, nda. 11 past 11 guys, we know 11 past 11, it all comes with luck and everything. So guys, here's the class. Tell your friends to tell their friends that guys, shosholoza shosholoza barini ikhuphukile isicathi ya yenzile lento siyagibela no stimela samalasha 1832 Mesida from Cape to Basic. Guys, tell your friends tiki mahala. Just subscribe, share with your friends, guys. Comment on a comment section. Like, you are allowed to criticize, to say anything that you want to say. Everything is free here. Just bring your refreshment, bring your water, bring your cold drink, bring your biscuits, and let's move on. Today will be fast nyana. Genetics and inheritance. Up until this time, up until this moment, guys, you understand what is a gene, what is an allele. Those alleles are being represented by alphabets from A up to Z. We did a follow-up yesterday. Go Facebook. So if you missed it, I uploaded the video here on YouTube. You can go and watch it, guys. So at this moment, you know what is allele, what is Mendel over drying. So today's topic here, monohybrid crosses. Monohybrid crosses, second subtopic under week 1. So okay, Reamover, as I was saying, or Shosholoza train, guys, from Cape to Messina, tiki mahala. Just bring your refreshment. Okay, guys, let's go to monohybrid. So monohybrid today what you'll be learning, the crossings of a single trait, like two alleles being crossed with other two alleles, and then you understand the phenotype. We'll talk more of phenotype, genotype. By this time you understand what are those, the homozygous, heterozygous, capital letters, small letters, the one that mask others, the one that is, you understand. And then we have to talk about purebred. The purebred kids two how can I say it? Purebred, the genes are the same, and then the hybrid, they are different. Like more hybrid example, ki horse and a donkey being crossed, and then rivaling liver a mule, ne? And then pure hybrid ki mutho le mutho, they have a baby. And then hybrid lo kry ki mutho and chimpanzee, and then lo kry ngwana, but that ngwana as ke hybrid. Okay, example, but the example of complete, incomplete, or partially dominant and co-dominant. These are 3 types of dominance. So guys, today we'll be focusing on this. At the end of this lesson, you should be able to differentiate between a complete dominant, incomplete dominant, and co-dominant. I agree, we understand that the dominant one kiyela ya capital letter, the dominant one and the recessive one. Here they didn't abadibeya. But we also have, remember, we also have a dominant and a recessive allele, and we said that these dominant alleles, they will mask these recessive alleles. You understand, like the capital letters and the small letters. So okay guys, monohybrid crosses, let's dive into our lesson. Yani amhela, we welcome you guys. Votanganedzwa. Yeah, you can comment on the comment section, guys, in your language. Swati, Zulu, Sotho, re a hamogela ntate wa rona. Zulu bathini Zulu when they say we welcome you. Siya ku bingelele to greet. Okay, hi, a re tswe ditabeng. Okay, monohybrid crosses. Monohybrid crosses king. We have to understand what is monohybrid crosses. Here we can get it from our baring. This one can mind the gap. Mind the gap. Yeah, only one characteristic or trait is being shown in a genetic cross. It means that we are going to have a genetic cross, but in this genetic cross, because we can have lots and lots of traits being crossed, when we cross only one trait, we say this one is a monohybrid. We are breeding one trait. Like for example, let's say hair color. That's one trait, Anir. And then let's say a cat, a cow's coat. Do you understand that coat? Ya home. That is a trait, Anir. So you might find where there's a father, and there's a father, and there's a mother. And then under that coat, at a specific locus, the father, he is having two alleles there, Anir. These two alleles, we say they are capital letters. And then the mother this side, she's having a small letters. Let's just say maybe this one they are coding for. By this time you understand these words. As I said, you have to understand them before we can come here, because here will be more explaining to code for. You understand this code, this alleles or this gene, it's coding for the color black. Ne? So we are going to say the color black, it's a trait, and the color black, if it's not black, it means that the cow is going to be white. So these are the traits, Anir. And then in these two, there's one that is dominant over the other. Ne? And then if the black is dominant, we are going to represent it by a capital letter A. And because it's dominant over white, the white is going to be represented by a small letter A. You get us now, ne? So these are the traits. The monohybrid, we are crossing one trait. It means that we are crossing one trait. If we are talking about the tallness of the plant, we are going to cross a tall plant and a short plant. If we are going to talk about the cat fair, the miririla, cats, we'll be talking about a trait. Ne? A whisk, the dilotsela tsa white, those things, the beard, cats. You might find that some cats they have long, some cats they have short. So those are the traits. Monohybrid. We also have dihybrid that we'll do next week. Dihybrid, it's when we are crossing two traits. Maybe we are saying a cow, it's having a black coat and it also having a long legs. Are they called legs? Yeah. Horn, big horn. Let's go there. The cow have a black coat and a long horn. Those are the true traits. Di will have dihybrid. Understand that, Amir. Monohybrid, we are crossing one trait. This is a genetic cross. We can say this is a genetic cross where we cross one trait. You are right, or one characteristics. You are right. You are very, very right. So, okay, examination guideline barry format for representing a genetic cross. So we'll have to learn a format for representing a genetic cross. Ne? So we have a genetic cross in this topic. In this topic, let's first, it will help you or it will help both of us to first understand this genetic cross, and then we take the genetic cross to a Punnett square. There is something called Punnett square that we also use it to demonstrate or to represent this genetic crossing. When we say a genetic cross, we mean the coming together of alleles or the coming together of gametes. That's cross. Ne? So okay, let's check for an example. We have a phenotype, genotype. Let's use a skin color. Let's use a skin color. We will say a black man, ne? A black man, ubilili kaka. They did they shared their gametes with a white lady. And then they will also tell you that the black color is dominant over the white color. Ne? Maybe, and then they said use a letter R to represent your gametes, or to represent your alleles, or to represent the traits. So here's the thing. The black is dominant, which means that because they said letter R, we have to use letter R. The black is dominant. We come here and say dominant R. And the white, because it's recessive, we are going to say small letter r. By this time, we know that this white r, because the white r is recessive, we have to have two small r for that phenotype to show. It means here we will have small r and small r for this white because it's recessive. Here in the black side, we can either have R or RR. Because the R is dominant over the small r, it means that the R here is going to be represented as our phenotype black, right? And then maybe they say, maybe they say this is the statement that they gave us, and then maybe they say, show us that there's a possibility for this black man, or this black guy. I've said this back there, that this black guy, we can say is brown, is yellow bone, is black. So they will say, maybe they say, show us a possibility that this black guy and this white guy and this white girl, sorry, this black guy and this white girl, they can have a white baby, not a yellow bone one. So it means that this black guy, her homozygous dominant. Yes, the lady, she is homozygous recessive. But because the guy can let's show urbakaba lingwana wa likuwa kiri. That means that this homozygous option I cancel. We are going to use this small r. Why, Mr. Paper? Because for us to have a small white, I want you to come with me here. This is the cross that we are going to use up until dihybrid, up until a pedigree. So you have to understand this. Why, Mr. Pip? They are saying that this white lady here and the black guy here, they are going to have a white child. For now, we understand that for us to have that one child, it means that she have to get small r from the mother. Let's do like this, and the small r from the father, so that she will be white. You agree with me, Anir? So it means that this small r merged with this small r and give us this. Ne? And then here's the thing again. As you can understand that this small r merged with this small r give us this small r. There's other possibilities. If bakabali bana ba bangwe, now why other possibilities that these people, they can have a baby and the baby might be black. Ne? It's when this R here, if this capital letter R is a sperm again. Sperm is if fertilize one of the egg this side, we are going to have a capital letter R and a small letter r. And this baby here, because we see the capital letter R which is dominant for the color black, ngwana utloba black, uzoba mnyama. Maybe akaba colored. Like some of them, they don't want to be called colored, but this is the matter of the truth. That child akaba colored. And then this is the cross that we are going to use, guys. This is the crossing. When we say crossing, we are predicting, or we are using Mendel's experiment, or the understanding of our genomes and everything, to come up with possibility. This R can merge with this R during sperm and the egg, and then we have this. Or this capital letter R, it can also go and merge with the second small letter r from the mother, and we will also have capital letter R and small letter r. Baby can be black. These are the chances, Anir, or colored this time. Colored, we write colored again. And then also we talked about the small r joining with the small r, or also the small r joining with the second small r, giving us r. So this baby will be white. So in this family, interracial relationship about nice a black guy dating a white chick. So in our family, the moment we see a white girl being born, it means that she's cheating with another white guy somehow. No, it means that I carried a gene here, a white guy. I get it. The white guy came here in South Africa, and then they enslaved. In Africa, enslaved our forefathers, and then they forced them to sex and raped them, and then after that we have coloreds. After coloreds, the genes were shed. This is what we are learning. After the genes were shed, and then now we are carrying those, because the color black is dominant. We carried the dominant maybe from our father and the recessive one from our mother, and now here we are. We are black here. We are running with these genes. And when we meet a white girl here, we find a white baby, it becomes a problem. This will do you understand? So guys, these things are important when you come to this. Okay, it's fine. And then in this family, we can see the possibilities for if we are saying the possibilities akere, if we are saying we are going to have 4 children, maybe re ba le ngwana wa mathomo, ngwana wa bobedi, ngwana wa boraro, ngwana wa bone, these 4 children, there's a possibility that 2 of them they can be black and 2 of them they can be white. So ratio is 2 out of 2. Then we bring this one to the simplest form. It will be 1:1. So now I've explained many things here. I've explained many things here, but also I want you to understand this cross, this is what is happening. These two here, they will undergo meiosis. When they undergo meiosis, the capital letter R will stay outside, and then the small r, and then small r, and the small r, and then now they will come to fertilize and then joins. We are fine, right? So, okay, we move there from genetic cross. What is genetic cross and the formation for presenting a genetic cross? We also has a Punnett square. A Punnett square. A Punnett square is this, what is happening here, same as what is happening here. And then where are we going to write a Punnett square? Let me write it here at the corner. A Punnett square is something like this. I agree, you are going to learn this more from the details. I agree, you are going to learn this with more details. Let's do like this. This is a Punnett square, ne? And then here in this box we will write gametes. We won't see this. We won't see this. Let me just find another space. A Punnett square, you'll see it, and I don't want to cancel this because I also want, after explanation or after the class, after this you can screenshot here, and then you will place it somewhere, or you will join them with you there. Okay, it's fine. Punnett square. Let's move on. We are also going to do the same thing, and Punnett. We call it Punnett. That one is a crossing, and also they can refer to crossing. And even if you write a Punnett square, I think black. I don't think they have that much problem. And then black, we are going to say BR or BB. This time let's do BB. RR. Capital letter R, small letter A, man. You see the mistake that I was doing previous. Small letter R. So in Punnett square, this one will undergo a meiosis, and then we will have R, small letter R, ready to mingle and ready to join. The possibilities now, they are there. The possibilities now are there. So in Punnett square, we draw a square like this, and then here we write gametes, and then gametophyte. Okay, it's fine. Then we write like this. And then mother and father, RR, we go and say capital letter R and capital letter R. And then mother this side, small letter R, small letter R. So here's the thing. I told you last that we have a rule, we have a rule of thumb, rule of thumb, ne? Rule of thumb, Kuri, you start with a capital letter. We start with the dominant one. So it means that in this square, we are going to join this R, we are going to join this R and this R. So here we are going to have capital letter R, small letter r. In this square, the following one, we are going to join this capital letter R and this small letter r. So we are going to have a capital letter R and small letter r. And then the bottom one, we are going to join this small letter R with the capital letter, like they are joining, like it. And then here we'll have capital letter R, small letter r. And then we are going to join these and these. They are going to join here. Capital letter R and small letter r. We start with capital letter and then we are going to have R. So these are the genotypes of the next babies. These are the possibilities of our next baby. You are going to learn this. We did F1 generation. So this one get the possibilities if we are going to cross them like this. So this one here, it shows that if we are going to cross a homozygous dominant with a homozygous recessive in that family, if I'm dominant, if I'm homozygous dominant black, if ngizimnyama zenkani zamagebla mhlaba wamasipala wahlala ngedoti, and ke nyala likhuwa, and that likhuwa it's recessive, and likhuwa le ke recessive akere ke yona isa this one is recessive and this one is dominant. It means that in our family, since we can see the possibilities here, the R will going to be dominant. We are going to be, all of us, we are going to be black. And then this color black, we say it's a phenotype. And then the genotype will be capital letter R, small letter r. So you get me, ne? You learn about this, don't worry. You learn about this. I'm just trying to give you another understanding of things before we can dive into deep into the real thing, or the putting everything into answering a question. So okay, it's fine. Let's move on. We can move on to Mendel principle. Then Mendel principle, Rebecca segregation. We talked about this again. When we see the principle of segregation, we are going to see separate two factors. These are the key words in segregation. Same as dominance. Here you can see they only concentrated only these two principles. And then law of dominance, we are going to see that the one is going to be dominant over the other. The one is going to be dominant over the other. Those two factors. And then in segregation, those two factors, they are going to be separated so that the gametes will contain only one of these factors. If I have R and R, it means that this R and R, these two factors, they are going to be separated so that the gametes will have one of these factors. You get me? This is the law of segregation. The question might come into the chooser, they choose A and B, or one, or choose from A to D, or one word answer. One word answer where they say an organism possesses two factors with separate of segregation to each gametes what one, then the answer will be a principle of segregation. Then that one we talked about it. And then let's move on. Then now we have types of dominance. And here we have a dominance that, there's a capital letter and what. Before that, we have capital letters and the small letter. And then we also have types of dominance. We have three types of dominance. Here they can say, give us three types of dominance, or name three types of dominance. We have a complete dominance, incomplete dominance, and co-dominance. I agree, we have capital letter A and small letter a. So when we find a situation where this capital letter A is totally dominant, like one allele is dominant, or we can say one allele is total. One allele is total dominant over the other recessive. It means that in this situation, if we are saying the A is coding for the color black and the A is coding for the color white, if we find this situation, the offsprings will always be black, because the A is dominant over the recessive. And we have a complete, this one is a complete. And then with complete, let's dive in here. Let's dive in here. With complete dominant and barini effect of the recessive alleles masked, it means that it will be shadowed, like we won't see it. If someone can be like this and someone can be like this, this individual, they might have the same traits but with different genome, because this one is masked. You understand? It's not expressed. It's not shown on the phenotype. You are black, but you have a genial who. Maybe you can be colored, but you understand with all colors they are black or they are brown, but we know in their genes they have small this one. Or this one, it might be just like me, black but having color, yellow colored. Okay, my color my yellow bone. Colors, they are yellow bone. When we say colors, when you and then you become yellow, ubali colored. You are mixing black and white, like your color is being mixed. But naturally, they are born brown with yellow bone, skinish color. Those ones, it doesn't mean that they have a traits like this from, no. It just means that the color that is dominant, it's A. The black color, the melanin thing. You know that for you to produce that color, you have to have a melanin. A melanin will come. Okay, it's fine. Let's move on. And then from complete dominant, we are going to have four types of dominant. Four types of dominant. Number one, a complete dominant, we can have a homozygous, a recessive. We can have a homozygous dominant AA multiplied with a homozygous recessive, small letter a, small letter a. Number two, under complete dominant, where we can see this dominant one color being dominant with another color, we can have a heterozygous mixed with heterozygous. My pen, heterozygous. And then number three, we might have a homozygous dominant, something like this, crossed with heterozygous. Or number four, we might have a homozygous recessive a crossed with heterozygous. So these are the possibilities, or these are the four types under complete dominance. When we see that when this A is coding for the color black, we are going to see more dominance over the small alleles. It means there we'll have this one, and then we'll have capital letter A and small letter a. And then here there's a possibility of this A and the small letter a, still the person will be black. And then here we'll have also the black color, and also we have the black color. It's when one allele is dominant over the other. And then let's talk about when neither of them they are dominant. Let's say, for example, neither of them they are dominant. Let's talk about a cow. We see cow. We see some of cows. They have brown. They have black. They have black coat on it. And then some cows they have white coat. And man, I'm drawing into Kisadiboni. And then this one is neither one of the two alleles of a gene is dominant over the other, resulting in an intermediate phenotype in a heterozygous condition. We have a cow that cow is inally black color, and then we have the other one inally white color. But when these two, because the black color is not dominant, let's not use black color, brown color. Let's use a brown color here. We see these brown cows and what. And we see that this brown color is not dominant, and this white color is neither dominant. When these two mix, they have a baby with another color. I'm not sure if you understand. We'll use example more. They will use example here, a flower. They will say a pink flower was crossed with not a pink. Is it a red flower? Sorry, excuse guys, excuse. They will say a red flower was crossed with a white flower. Neither of them they are dominant over one another. So it means when I take this red allele and cross with this white allele, the color that I'm going to get is another color. Might be pink, primary colors. So it means if I'm crossing red and blue and blue primary colors kingling, blue, yellow, red. If I'm going to cross a yellow flower, a red flower, it means that the color that I'm going to get, it over mix, because neither yellow is dominant, neither red is dominant, and then the primary color will be yellow and red. We are going to have orange. We are going to have orange flowers. So we know that when we see two flowers being crossed, and then neither of them there, that one is incomplete dominance. We can see that this color, okay, and then when we cross it, example, when we cross it, we cross this with capital letters, because neither of them they are dominant and neither of them they are recessive. Ne? So we are going to say a yellow capital letter Y mixed with capital letter R. Do you understand? So yes, now more colors come in. Yes, now more alleles come in with different why don't we write capital letter Y multiplied by capital letter Y? You understand how now again, because this one is a yellow and this one is red, and this yellow is not dominant over the red one, and the red one is not dominant over the yellow one. So it means all of them, they are just a half full. Yeah, let's just say half full. They are not longer the small letters, but they are growing to be the bigger letters, but they don't fill up the cup. If we are saying we are going to have a cup here, and then this cup is yellow and then this cup is red. They are not filling the cup. When we talk about the dominant and recessive, this one fills the cup. The dominant one is going to fill the cup, and the recessive one is going to have our half here. Hence, when this half and the another half mix, they become that yellow color. But when this, let's just say maybe red is dominant over yellow. When this red here mix with the small, we already have the full one, which we're going to code for the color red. I'm not sure if you are taking this, but let's move on. And then we also have a codominant. Codominant is when both alleles of a gene are equally dominant, whereby both alleles express themselves in a phenotypic in the heterozygous condition. Let's talk about this now. Here we have a new color in the incomplete dominant, they resulting in an intermediate phenotype. This is a keyword. This one is a keyword to differentiate between these three. Incomplete dominant, intermediate phenotype. So we are going to have a new color. Intermediate means middle, center. We are in the at the intermediate. We are cutting it into half. So we are going to have center, and then when we cross this product, maybe like we are having a pink, we are going to have a red and red multiplied by white and white, and then RW, this one is an intermediate color, which is pink. And then if I were to cross this pink and another pink, we can see now we can have red and then we can have white. Now we understand the joining and the interjoining. And then codominant is when both of them they are what? Both of them they are dominant. So in other books you'll find that they represent, let's come here, don't have space here. In other books they might say they might represent them with C and capital letter. I'm not sure. C for codominant. Why? Because we can give you this statement saying a codominant alleles or a codominant a family. Codominant. Okay, let's explain codominant again. Codominant is when let's take example. Now we have a black cow. This black cow is going to be mixed with a white cow. Black cow is going to be mixed with a white cow. Codominant, and a codominant black cow. And then because both of them they are dominant, they are equally dominant, we are going to have a cow having a dot black and zebra. Not zebra, man. Zebra is something else. A cow with a black dot, or a black cow with white dots. That shows that no, that cow is codominant here. It's very both. It means that if we can take that cow, more it's enough farming. If we can take that cow and cross it with another cow of that nature, they will give us a white cow or a black cow. Why? Let's say here, black color, it's BB. Both of them they are codominant. When they say both of them they are codominant, or both of them they are equally dominant, it means that we are going to use different letters, but with something like this. And then when this B join with this one, they form a new. Hence I told you that some books they use CB multiplied by CW or CW to show that this color now it's CB and codominant W. It means with this codominant, the color here is neither black or it's neither white, it's black and white. Black and white equally. Both alleles express themselves in a phenotypic in the heterozygous condition. And then you learn this, we understand this. We have already at 35 minutes, and then when we go that side, we'll be pushing 2 minutes, 10 minutes will be done. And then by genetics problems involving each of the three types of dominant, we'll have to do problems involving these types of dominance: codominant, incomplete, and complete. Then we can also will do this, and then they're saying proportion and ratio of genotype and phenotype. More when we work up proportion and ratio, that's what I was saying here, that here we are going to have genotype ratio, it's 100%, and all of them they're going to be RR, and the phenotype is going to be 100% black. Then yeah, then we'll use that ratio. If we are saying we have 1:2:1, and out of 4, it means that 25%, 50%, 25%. If it's 1:1, it means that 50%, 50%. Then if it's 100%, it means it's 100%. So okay guys, let's move on. Re tsena ko the question paper, the questions and what we can pass by our class, our textbook. Law of segregation, separation anir. Here two factors separate. We claim it like that. Dominant, one allele or one factor is dominant over the other. And then assortment, this one they don't ask much about it, but this one during metaphase, but what chromosomal random arrangement that make sure that one gametes have different alleles. Okay, it's fine. Genetic diagrams, a variation, it caused variation up there. Genetics diagram, but it is important to start with a pair of alleles in the mother and another pair in the father, because each individual inherits 2 alleles for a gene, one maternal and one paternal, the bivalent chromosome. What is a bivalent chromosome? These are the chromosomes that they are kissing and touching. And then this pair may be identical, homozygous, or different, heterozygous, we know. And alleles are represented by letters, we know. Capitals for dominant, we know. Small letters for recessive, we know. P stands for parent generation. P stands for parent generation. We are going to learn about this. Let's go now. They say F, we have P there, and then F here, it stands for filial generation. But remember, F4 family and F1 generation is the first filial generation and F2 is the second. But when they want the first generation, this family generation, F1 generation, you can get it wrong. Filial generation, because family generation is just the F rhymes with family. Doesn't mean that, okay? So here's the genetic cross that I was talking about. But it is important to learn the exact layout, because marks are allocated for the layout as well as the correct working out of the cross. Use the following genetic diagram or template to solve all genetic crosses. So guys, okay, we are left with 23 minutes if I'm not mistaken. So we can do this, guys. Okay, it's fine. But layout of genetic, this one is the genetic P1, but get parent again. Parent, and we can say parent here, parent 1. And then let's say for the example, we can add what I was talking about. This is what we have to write it, or this is how we have to present it. Our example, yeah, a black man, they said we have to start with female. Yeah, they said we have to start with female, but now I also prefer to start with male. But now they said we have to start with male. Doesn't matter, as long as you place the phenotype, the one that they will say the 2 parents, one is homozygous what, and the other is what. That first person, if they say 2 parents, the lady, the mom, she's homozygous what, and the father is what, that's what we take and place it here in this group, in this chart of us. Then we will write P1. You'll come and write P1, and then after writing P1, you'll write phenotype, and they will give you the scenario, or maybe a black guy. In this scenario, they'll say a black guy went to England, and when he arrived to England, he find a wife. They're not going to, you'll see how they ask question. I will also go to one of question, and you see how they ask it. Black guy, and then crossly a white guy, again, white person, and then they said a homozygous dominant father. But use letter. Maybe they will say use a letter. Which one we haven't used that can fit well? A, B, C, D, E, F, D. Let's use D. Use D for dominant, and for dominant, and maybe they can say D for dominant. It means that small letter D for recessive. And then they say the black guy is dominant over the white guy. You understand? When they say that, it means that we are dealing with a dominant, a complete dominant. Ne? We are doing with a complete dominant. And then we'll come and say phenotype. Phenotype is what we see. We are seeing our guy being black. When now you'll write from phenotype, you'll write P1 and you'll write phenotype and you write black and white. Ne? And then from there, genotype. Genotype, the dominant and what and then they said homologous. They gave you as instruction about a homologous black guy. Because they said homologous, it means they're the same, capital letter B, with a homologous recessive white lady, small letter B, small letter B. And then we come here. You write meiosis because the gametes, they will undergo meiosis, and after undergoing meiosis we'll have gametes, and then we'll have this B and B separated, and this side we'll have small letter B and small letter B separated. You see now, they are giving you the marks here. And then at the bottom here, they also said P1 and F1 meiosis and fertilization. They have marks if you have placed them at the correct position. This meiosis and fertilization. Meiosis between genotype and gametes. Ne? And then fertilization will happen when fertilization happen. This B this B will come and join with this small letter B here. This B will come and join with this small letter here, and we will have capital letter B, small letter b. The F1 generation now. This one get the babies that possibilities. And then this B here can also come and join with this B, and we'll have capital letter B, small letter b. This capital letter B possibilities, capital letter B, small letter b. And then this capital letter B possibilities, capital letter B, small letter b. And then this one are the genotype of the F1 generation, not the offsprings. This one get the offsprings. And then the phenotype, if we have this, we can see we have the dominant one and the recessive one. Here, because the dominant is going to win, the color will be black. And then here the color will be black. You write everything. You even write the phenotype here, because you can't just come here and write black what? Black phenotype. Then you get your mark. Okay. And then they may ask you, write the ratio. Here at the top, ratio is 100%. And then here at the bottom also it's 100%. It means anyhow, whether they are going to have sex up until they die, those people, they are going to give us the offsprings that are black. We want to have a white baby there. So okay, it's fine. From there, we have an explanation. Come on my side, I've already explained it for sure. You can go and write it and then read it as your notes. And then here's the Punnett square that I was talking. You can write that one, or you write this one. Here's the or. You can use that crossing, but if you feel like I don't understand that, now I want to understand, I do understand this one here, square. This also, we call it a cross. But maybe they can bring a question that write a Punnett square, you will have this. Or a crossing, you will write that one. But if they just say genetic cross, if you write this or write that one, totally fine. Then you will also come here and say P1, the phenotype, you will write black, same, and then you will write white. Same thing. You have to place this because there's a marks for this. You don't just come and write your Punnett square here. And then the genotype, you will give us the genotype, you will give us the genotype, and then the gametes, the genotype will undergo meiosis, and then meiosis, the gametes will be B, and then here small letter b, and then fertilization will happen, and then we'll have the gametes here, the father and the mother. The father going this side B, or the first one that we will give instruction to, homozygous or heterozygous someone. And then this side will have small letter P, small letter B, and then this B is going to cross and join with this B here, capital letter B, a rule of thumb, and then this B and this B, capital letter B, small letter b. We start with capital letter, and then this one join here, that one join here, B, and then this one come here, and this one come here, B. And then we come and copy here the phenotype here. It will to make a genotype. Here we have to start with genotype, guys. Exchange. Let's exchange this so that the thing will be able will be the phenotype will be explaining the genotype. Genotype, we are doing like this, and genotypes we are going to say this our genotype, copying them here: B1, B, HB. Then ne, don't forget here genotype B, and then the phenotype we will be explaining now. Then here we'll write phenotype, then this phenotype, and then here you'll write genotype. This phenotype will help you to say I'm explaining what is up there. Big will win, black, 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 black. Then you get your mark. And then if the F1 and the P1 they're at the right, we get 1. And if the meiosis and fertilization are at the right place, you get 1. So this might come out of 7 or out of 6. So you know that you have 1, 2, and 3 for crossing, and then 1, 2, 3, 4. Then you get 1 from here. The compulsory one, there's a compulsory mark that comes with this. Maybe they say black and white, find give us or do a panel square or do a genetic cross to determine the genotype. When you determine this genotype, that one will be your compulsory mark. Or if they say show us the phenotype, when you show that phenotype, that will be your compulsory mark. If you don't have that mark, it's wrong. Everything might be wrong, or you might only lose that one mark. And so it will depend on the marker. If when you just crammed this BB, CC, or you understood why were you doing this BB and CC, you get me? So if the markers sit down and decide that when you were just you crammed or speaker, you're cheating, hence you managed to come up with this crossing and everything, but you failed to give us the answer. That you gave us the work, but you didn't give us an answer. So due to you not giving us an answer, this compulsory, if it's wrong, everything might be wrong. Or we might say, let's give that person 6 marks and then he will lose the compulsory one. So it will depend there. So guys, from there, very important, you may also be asked to work out the ratio or percent chances of the various phenotype or genotype occurring. So if there are 4 possible gene or phenotypes in total and only one having a particular phenotype, it will be 1 in 4 ratio, 25%. For an example, we find it when we cross a complete dominant, when we cross a heterozygous with a heterozygous, ne? And then we're going to have capital letter H, and also capital letter H, small letter h, and capital letter H, small letter h, and small letter h, and small letter h. So it means the ratio, we're going to have 1 capital homozygous dominant and 2 what? 2 heterozygous, and then 1 what? 1 homozygous recessive. This one ke ratio ya genotype, and then in phenotype we are going to have 3:1, because these 3 they are going to code for the dominant one, and then 1 is going to code for the recessive one. So here, if they are saying a percentage, it means we are going to divide percentage. We divide by 100, and it means here we're going to have 25% plus 50, which will be half, and then plus 25%. So for this one, it means we are going to have a 75%, 25 plus 25 plus 25, and then 25%. So if, for example, they say we are going to have offspring, 100 offspring, and then in this 100 offspring, how many of them are they going to be recessive, or to have this color that is recessive here? It means that 25 of them, there's a possibility for them to have, since it's percent, it would mean that 25, there's a possibility of this 25 to be a recessive one, but the 75 of them, they can be dominant. So this is what you will be asked they will need you to understand. Ne? I hope we are understanding here. Yes, they said 1 in 4, it means 25% chance. I've explained it. But you need, I left with 10 minutes. You need to read the genetic question with great care. Go and read this. The phenotype that they're talking about. Are they talking about the seeds? Are they talking about the flower? Are they talking about anything? And then the dominant one, which one is dominant between the two? And heterozygous or homozygous. Are they heterozygous? Are they homozygous? And the letter that we are given. Ne? We have to take care of that. And then monohybrid crosses. We talked about these three of them. We have complete, we have incomplete, and we have co-dominant. And then Friday we'll do sex determination and sex linked grouping next week. So here we deal with this. And then let's move on. Monohybrid crosses with complete dominant. We say that complete dominant, we have here we have 4 types. And it can either be homozygous multiplied by recessive, and we can have 4 types. We talked about this, let's not waste our time. And then here's the genetic problem number 1. But seeds can be round or wrinkled. Complete dominant again. It's a complete dominant problem. But use a genetic cross to show the genotype. To show a genotype, it means at the end of your answer you will have to give the genotype. Use a genetic cross to show the genotype and the phenotype of the F1 generation when 2 heterozygous different plants with round seeds. Round seeds is the dominant. Heterozygous plants with round seeds are crossed, but the allele for a round seeds is dominant. Ne? The allele for the round seeds is dominant over the allele of a wrinkled seed. Use the letter R. What if you let the letters and the small letter R? Ne? So here we come. We say P1 phenotype. They said a round seeds. Where do we get the information? F1 generation when 2 heterozygous plant, 2. This means we are 2 heterozygous plants with round seeds. The phenotype of one is round seeds plus a round seed. That's the phenotype again. These are the 2 heterozygous round seeds. It means that we know that now they are heterozygous genotype. They have capital letter R and capital letter R for them to be heterozygous, for them to be different. Ne? From here we are heading straight to a question, so that you'll see. Then you'll read for yourself now. Then we have meiosis, we have gametes, and then after here, when you got this and this, there's downward slope. Wathelela mo. Then you have a capital letter R, small letter r, the gametes, and then they will form this one and this one. And then phenotype will have the round seed because of the dominant one, round seed. And then because this one they are small, they have a wrinkle seed. Or you do your panel square. Panel square, the capital letter R and the small letter r, they will join. Capital letter R and capital letter R, they will join. Small letter r and the capital letter R, they will join. Small letter r and the capital letter R, rule of thumb, the tomaka capital letter, and then small letter and the small letter, small letter. And then here, if we were to give a ratio, but genotypic ratio of this one, 1 RR, 2 R, capital letter R, small letter r, and 1 is to 2. I did example cut this. And phenotype 3 is to 1. We did example with this one again. We are moving. And then we can explain all crosses between 2. Explanation, there's a cross between 2 heterozygous plants produce 25 homozygous dominant and 50% heterozygous and 25 homozygous pair offspring. Then there we have homozygous with a complete dominant. Again, is another exercise. And then barfilets onaka at the color. This one is a homozygous yellow and homozygous green. Green line. Then it means that if we have homozygous, it means they're the same. We might have yellow color. Which letter did they give us? But a Mendel cross homozygous plant with a green seeds with homozygous plant with had a yellow seeds and found out that all offspring were yellow. Okay, all offsprings were yellow. I've constructed a genetic diagram using to show the F1 and F2 generations. Cross, if you choose a letter, okay, you see here they are giving us an option to choose a letter. Choose a letter. No, to use one letter capitalized for the dominant and the small or lowercase for the recessive. So here they give us a yellow and a green. So we are going to have one letter. Let's use green, G. Ne? So it means because G is dominant over the small letter G, it means that because all of them they were yellow, because they told that man, they said had is female plants with a green seeds, a homologous plants with green seeds, uh with homozygous plants with which had yellow seeds. Barenge. Mendel crossed homozygous plants. You see now, Mendel crossed homozygous plants. They are the same, and with green seeds. Ne, it means green here, this side. And then with homozygous plants which had yellow seeds, this side, and found that all the offspring were yellow. It means that the offspring, oh yeah, man, here's the thing. I was being confused of this offspring here. Oh, and then yeah, let's go back. Let's dive in. And then it means that because this and this, the all F1 generation, they were yellow. It means here we have capital letter, and then here because it's homozygous, small letter, and then after crossing we are going to have capital man. You see, I'm also doing mistake. And then after crossing we are going to have capital letter G and a small letter G, and then capital letter G and a small letter G. Ne? G being coding for a yellow dominant. And everybody choose any dominant. Choose any letter. You can choose V, you can choose, but I rather choose those that when the capital letter is more different to the small letter. For example, if you are going to choose Z and Z, there's a possibility that your handwriting is bad here and we can't see which one is dominant, which one is not. So okay, and then here after crossing we'll have capital letter G, small letter G, and then capital letter G, capital letter G, capital letter G, and then this one, and this one, then this one and this one, capital letter G, small letter G, this one and this one, capital letter G, small letter G, G and G, small letter G, small letter G. You get me? Then here 100% yellow, and then here phenotype is 3:1. Ne? And then let's go on. And then another activity, you will do it kabuena, and then incomplete dominant, you will do it yourself, that one. Incomplete dominant when two, I've explained this, and hence I'm cha. Incomplete dominance cross between two. This one, they have a new phenotype. That's what we have to get. And then here's the example. They're saying a homozygous, it means same red flowering plant crossed with a homozygous white flowering plant will produce plants that have a pink flower. The moment you see one color results one color, you know incomplete dominant. And then the phenotype will be red, the capital letters, and then Punnett square, by very clear Punnett square, and then this will match with this, and then all of them they will be pink. Then all of them pink phenotype. And then from there's another example. Look at the flower. We have a red flower, and then we have a white flower, and then this one will cross with this one, and then we'll have a pink. And then this gamete will also cross with this, and then we'll have pink. And then the new genotype, it's RW, and the new phenotype, it's color pink. You understand this one? And incomplete dominant, we understand this one. And then incomplete dominant, here's the activity 3, and then this activity 3 will do it tomorrow. Let's do this tomorrow, and let's do this tomorrow. And then codominant, when we have now they say codominant, both of them, they are equally dominant. Then red coat, white coat, cow here. In cattle, three color variation are possible: red, white, or a red, white, or red and white. In patches, Riwaka codominant. This, you see, then that RR for red, and then WW, and then you match them, you match them, you match them, you match them, and then you have another new genotype, which is RW. But because it's a co-dominant, all of them, they are all red with white patches. Here's an example. We have this color here, and then we have this color here, and when we mix together, we find a color which is co-dominant. Means here we have red, red, multiply with white, white, and then here we'll have RW, which will be red and white. This one is complete. So this complete and incomplete thing. You are done, my friend. So that is from me, guys. My time is up. I only have 1 hour with you guys. So okay, that was it. Next time we'll do sex determination and sex linked. So yeah, codominant. Note, inheritance of blood groups is also partly an example of codominant when the alleles. We are also going to do this blood grouping next week. There, but they said this will be dealt later. Babu need. They are not wrong here. We'll deal with this later, not now. So, refitamo. We have 3 types of dominance, or we have 3 types of monohybrid: codominant, incomplete dominant, and incomplete dominant, and a complete dominant. So you at this moment this is what is happening. So let's go and do this, guys. Zamang will work, and then after working it, we'll come and do it Tuesday, doing our live video there at Facebook. So Yanko Facebook, guys, African transcribers, follow, guys, subscribe here for more, comment, share, guys. Don't forget to share, and let's guys. Let's give that thumb up so that these videos can be they can be suggested to other grade 12 students, or for other people who are looking for this information. So from me, guys, thank you. I also had a question paper, this one, 2018, ne 2018, this one. Let's go and do 2.4. So tomorrow we'll do 2 correction. From there, I know when I come with life, I'm only there to do correction, and after doing corrections, we move on. 30 minutes of that live videos, and then we'll also do this. I see that I don't have time now. This is what I was planning, that I will tackle it. 2018 question paper, November 2018, November, paper 2, question 2.4. So guys, I'll screenshot this and send it to the WhatsApp group, so that you guys, you'll be able to do these 2 activities. Thank you. Until we meet again, that's it from me, Mr. Mpepu. Welcome for thank you for tuning in, guys. Don't forget to subscribe, don't forget to like, guys. Don't forget to share with other friends. If you feel like you can't understand me, I'm fast, I'm slow, or I'm just saying things non-stop, I don't have full stop, I don't have what, you can share this to other learners. You might find that these things, they are going to be useful to someone, guys. Let's share it. Just share it and leave it like that. It's just like it, or just comment that grade 12 hashtag grade 12 class of 2024. Let's help one another, guys. Ticket is free, subscription is free. You listen, you attend, you don't attend, totally up to you. So thank you, guys. Welcome, and then let's head to our distinctions at the end of year. Thank you. See you again. From me, signing out. Peace. We'll do this tomorrow, ne? Don't forget. And the school are closed, so we might do this around 4 5. because also I also have a problem with a network during night. So yeah, stay tuned, guys. Thank you.